it going, everyone? I'm back in action. I got my light set up in a place where it hopefully won't fall. And you can see me a little bit better in my lopsided dress. But anyway, I made some dinner, and I thought maybe y'all could keep me company, or we could eat together. Maybe you want to grab some of your food. And I know some of you like to know my recipes that I just make up. I just like make it up while I'm making it, you know, because then I guess it feels like I'm a wild animal, just like foraging for food. And then maybe it empowers you to be like, oh, I have those things in my fridge. I don't have to order in takeout or go to fast food or anything. I could whip something up that's nutritious for me too. And that's what we're going to talk about. We're going to talk about nutrition. Speaking of which, you don't need to wear socks all the time. Let the toesies breathe. You know what I mean? Oh, I think I see a comment. Hey, Jason, what's going on? Are you eating dinner now? Do you want me to inspire you with some delicious, nutritious food filled with vitamins and minerals? I can't wait to show it to you. Let's see. I am going to send this link, and then I'll be right back. Oh, Garland is in the house. Okay. Here we are. So, before I talk about, you know, Shilajit, which is legit shit. Okay, Shilajit, I want to have a conversation tonight. Besides the food that we're eating and rejoicing with, blessings on all of your food, on my food, on the people that grew it, prepared it, packaged it, all that stuff. Bless you. Thank you for taking care of this sustenance for us, you know? Oh, sacred place we live in together. Besides that, I want to talk about Shilajit. I want to talk about m6A. I want to know if there's any doctors out there that, obviously scientists, you know, way more than me, but I'm geeking out on it, you know. I want to talk about aflatoxins. So we've got a lot to talk about tonight. Are you in for it? Are you in for the conversation, or what? You want to talk about Atticus? Atticus says hi. And Zuriel. Oh my God, those are gorgeous names. Thank you. Okay, so y'all want to know what's going on here? Okay, I have my to-go container here because I'm not going to eat this all, but I didn't want to leave it on my stove in the other room. And I've got my my like chamomile-ish tea. Chamomile-ish. Because I don't know what else is in it. I forget. It's like an herbal blend that I got from Lidl, which is like $1 something and organic, they say. Do y'all trust organic label? I just don't know anymore. Anyway, here we are together on a Friday night with beautiful weather. You had spinach fettuccine? So was it like spinach with fettuccine noodles, or was it like green fettuccine noodles? I remember the first time I had green noodles at my aunt's house. I was scared. I was like, are you kidding me? This is food? Where's my beige noodles? I don't understand. It's getting too hot for this sweater. Saturday night? It's Friday night. Oh, it is Saturday night. Garland. Oh my God. Well, I could pretend I'm talking about my last night's dinner, which maybe I should share about as well. But I didn't have dinner last night. That was one of those rare nights when I had blueberries for dinner. I don't know. I don't think I was hungry. I obviously had food, the same food that I made today. Okay, so today, are you ready? There's so much food on your plate. I can feed 5 people with this. But seriously, It took a little bit longer just so that I could put a couple of cucumbers on my plate because I'm obsessed with cucumbers. So good. I have been having fun. Also, I don't want it to be... Time is moving too quickly. Like, I want it to slow down a little. I have an event next week. I have so much to do. I can't do enough in a day. Like, I'm going to be inviting people the day before. They'll be like, why didn't you tell me last week? I don't know. I was hanging out with you all. I just can't invite people all the time on time. It just doesn't happen. And I apologize because I want you all to be there. So anyway, I put some butter in a pan. And then I call my friend Stan. Not really. His name is Harry. And then I put 3 tuna steaks that were like this big. And then a pile of arugula on the side of it, just simmering in the butter. Put a timer on for 6 minutes. Waited for it to simmer a little bit. And then on one side of the tuna, I added a little bit of sea salt, fresh ground black pepper, a little cayenne, a little turmeric, that's it. And then I flipped it over after 6 minutes. Oh, I have this like garlic spice blend I put on it. I added more arugula. Like the arugula just cooks down. This is like almost a whole box of arugula. Put some string beans. Actually, I didn't even put the string beans in it yet. I put the arugula. And then a little salt on the arugula. And then a little more spices on the other side of the tuna. And then I got distracted talking to my friend Harry. And I splattered a little bit because it was frozen and it like banged in the pan. But that's okay. I didn't burn myself. And I started my tea boiling. Because it's nice to have tea with your dinner. Don't you think? It helps you digest. Oh man, I hate it though when my tea bag label falls in the tea. Ah! All those weird pesticides, all those weird inks that I'm drinking now. Don't drink the ink. Okay, what do you think? Okay, go to bed. Oh, your kids gotta go to bed. Well, I'll be on here probably for half an hour, so if they go to sleep, quickly come on back. Have you played them Scribble Soup yet? Oh, I gotta mail it to you. But Alexa now sings Scribble Soup songs. Okay, so where was I? Are you following this? Any questions so far? So then I flipped it over again because I didn't want it to be like rare on the inside. Like it was frozen, so it needed to cook a little longer. Okay, and then I added a little bit of heavy cream. I just like made a little spiral design over the whole pan. Added more arugula. Added some string beans. Added some broccoli. I had 1 piece of asparagus left in the fridge that I forgot for another dish. I gotta take my sweater off. Oh my god, it's hot. It is hot in these lights. This is show business, y'all. I don't know what's up with the crooked dress. And what else? Oh wait, and then finally smoked paprika. Oh my gosh. An amazing addition to like anything. I flipped it again and I chopped it up in the pan just to make sure it was cooked on the inside. And oh, I added lemon juice, fresh lemon juice, like chopped open a lemon. I didn't chop it, I sliced it and I squeezed it right in there. And that makes all the difference, people. That freshly squeezed lemon. Now, I know what you're thinking. I know what you're saying. I'm not going to cut a darn lemon. I'd rather just call takeout. Well, if you don't feel like cutting a lemon, what you could do is just buy that lemon juice in a glass jar, organic preferably, and then just pour it in there. So anyway. Oh, I ate on this before I came here. I'm sorry. I didn't keep it all fresh so you would see it, but I was hungry. Avocado, sliced on the sides, and I poured hemp seeds all over that dang thing because hemp seeds have so many nutrients in them. You would not believe. What? Am I boring you? Okay. We want to talk about what Shilajit is. Does anyone know what Shilajit is? Shilajit is like dirt. It's like really mineral rich dirt. And it also has fulvic acid in it. And fulvic acid is supposedly like it helps you absorb things. It's like fertilizer for people. You know, it's like compost tea for people. That's like what Shilajit is. It's like your fertilizer. Does that make sense? Anybody else know more about Shilajit than me? I just think we should talk about some Shilajit. It's legit. Shit. It really is. Okay. I think what I'm going to do is Google some readings and read them to you about Shilajit and m6A and aflatoxins. Now, you might know I have no idea what I was just going to say. Like, I really just don't even know. What was I talking about, Garland? Come on. Were you paying attention? Avocado. I think I learned the avocado cucumber works good with fish thing from the whole world of sushi. You know, the avocado nori cucumber roll. Yeah. So, okay, the thing, how do aflatoxins and m6A, like, why talk about that in the same conversation? Well, I think it's good to explore what is it like to modify our DNA? You know, this is something that's happening kind of for the first time. I mean, we've had GMO foods for a while. You know, it's hard to avoid it, especially soy, corn, and cotton. Those are the biggest GMO foods, I think. And the biggest risk, y'all know I wrote the song Clap for Food Quality, which is about, you know, be careful with GMO foods, basically. And that includes tilapia and turkey, by the way. Those are like these weird creations, like these weird modifications, I think. Like wild turkey is completely different than the turkey that you get on Thanksgiving. It's just like, I heard it's like genetically modified. Does anyone know about that? We need to look it up. We need to Google some stuff together and ask the experts, of course, before we export the information into our hard drive of knowledge, the brain. Anyway, thanks for being here with me. I love you. Thank you for hanging out with me. Why do I say Okay. So in the 70s, m6A was either discovered or became more of a thing that they found in people's bodies or in viruses or whatever. Aflatoxins also became a thing that people started to be aware of, which is basically like a mold spore bacteria thing. I think it's like Aspergillus flava something is the whole name. I'll look it up in a second. But what it is they found it on a lot of grain, like this like microscopic mold, and it was making people sick. And so now if it's found, then it's recalled. However, it's recalled for humans, but they feed it to animals. So if you don't get organic, actually, maybe they do feed it even to organic meat. I don't even know. But that's one reason I think why the vegan diet is so cleansing. It's not just about purifying yourself from meats, which I'm not preaching the vegan thing. I'm not vegan anymore. I think you just got to do what feels right for you. I'm just saying, like, that's one reason why I think it's so cleansing, besides the karmic, like, horribly way they're treated. There are so many, they're given like tons of antibiotics to deal with the fact that they're eating crap, and they would be getting sick every day without the antibiotics. So then you've got these bacteria that you can be eating in them that are resistant to antibiotics. Now this is industrial farming, industrial animal farming. Why is that necessary? So in industrial animal farming, I mean, I'm sure you've seen the PETA videos, and it's disgusting. I mean, it's just absolutely disgusting how they're living. The people in the neighborhoods have to smell like they're like spraying the Shilajit in the air, even though unfortunately it's not Shilajit. Or I mean, I don't know if you want to spray that in the air. But anyway, it's not good stuff. So the aflatoxins are the grains are fed to these sentient beings, these animals. And so then it becomes part of them. And then you end up eating them. And they especially find it in the milk, the aflatoxins, which supposedly, like if you boil it, it can kill some of it. But there's a list of different aflatoxins. Like one of them is called B1. We really should be having this conversation. We should be having the conversation about aflatoxins, Shilajit, and PFAX, which I talk about in Hempdemic, the plastic molecules that are in our drinking water that we bathe in that gets in our blood. They said 98% of us now have like plastic in our blood. So basically, like, long story short, it F's up our livers. And that's why I sing about the liver a lot in my music. Like, we have so much liver cleansing to do, because the heavy metals that get into our bodies from also the lack of regulation of our soil, and like how we're all responsible. We're all doing this. We're using power, we're using electricity, and the power companies, like, it's not, I don't think we just need to be like, you're bad, we're good. I mean, we're using the power. So let's help our power companies make better choices. Let's not be rude to them, you know? Let's figure out what to do with this coal ash together. Let's plant sunflowers around it. Let's plant cannabis around it. Let's remediate it. I know this sounds idealistic, but why not try? Why not try at least? I mean, we are sustained by food that grows in the dirt. Let's get down to the basics, people. Don't escape into the fantasy world of, I can just call a number and get food delivered me to me. I mean, yes, that's true, but like, let's not pretend that that food doesn't come from the ground at some point. We're all in this together, okay? So where does this mold come from? Where does the aflatoxins come from? Well, a lot of it's bad storage. Very prevalent in peanuts. So what's going on? Like peanuts and cotton and corn and grains, but peanuts, cotton, and corn are the top 3 aflatoxin issues. So I don't know if organic products are left out of this equation or not. But if you're going to do a few things organic, that's probably a good option. Like I cut corn out and it made a huge difference for me. Even organic corn chips, probably. I mean, if anything's in a bag, it's got something in it anyway. But I mean, who doesn't like crunching on things, right? Anyway, what do y'all think about this? Are you still there? Did I put you to sleep? Are you sleeping? I'm glad I can see the chats. All right, so I'm going to find a reading for you about aflatoxins. And then we'll talk about m6A. Oh my God, that's so good. I've always dreamed about opening up a restaurant. The definition is, aflatoxins are a family of toxins produced by certain fungi that are found on agricultural crops such as maize, which is corn, peanuts, cottonseed, and tree nuts. So peanuts and cottonseed are huge in Georgia. So I want to talk about this, y'all. What does it mean when you got a huge agricultural business in your state, which is wonderful? I'm so proud of Georgia for being able to supply this stuff. At the same time, what about the community? What about the water? What about our water supplies? You know what it is? It's in the air. The aflatoxins get in the air. Y'all, is that what this yellow pollen crap is in Georgia? Is it aflatoxins? We need to test that, Shilajit. For real. Who's got the lab? Connect. I need to connect with you. Okay. So anyway, it's definitely in the air, soil, and water. There's, mold, it gets everywhere, right? So these aflatoxins, these like nanotoxins, these microscopic toxins, they might even be on hemp growing, but I will say the more THC that grows out there in the sunlight in the world, those THC, those trichromes, they will trap the pollution out of the air. See, that's what we need. That's a solution to pollution. Don't fall asleep on me, Garland. Okay. The main fungi that produce aflatoxins are Aspergillus flavus. That's what aflatoxin comes from. Aspergillus flavus and Aspergillus parasiticus, which are abundant in warm and humid regions of the world. So we got them in Georgia. We probably got them in Texas. I think Texas has that. Texas has that yellow stuff too. Okay, Mama. Anyone here that was old enough before 1970? Can you tell me, was there yellow pollen in the air in Georgia or Texas before 1970? Oh, you're awake. Good. Because if there wasn't, then maybe the pollen has a little bit of aflatoxins in it, and that's why it makes us cough. Okay, well, anyway, it has been studied by the Cancer Research Institute, and it is a known carcinogen now. Aflatoxins are carcinogens. Why? Aflatoxin-producing fungi can contaminate crops in the field at harvest and during storage. How are people exposed to aflatoxins? People can be exposed to aflatoxins by eating contaminated plant products, such as peanuts, or by consuming meat or dairy products from animals that ate contaminated feed. Farmers and other agricultural workers may be exposed by inhaling dust generated during the handling and processing of contaminated crops and feeds. Now, I'm wondering if peanuts are exposed to it a lot because they're underground. Now, this is what I'm worried about, y'all. Roundup, I read something about how like Roundup creates like a mold resistant genetic thing, which actually makes it so that the mold ends up somewhere else on the plant. Hold on. We don't need to be using that stuff. Why are people still using that? So many people won the case about Roundup causing the lymphoma thing. Anyway, glyphosate. The glyphosate, the aflatoxins. Ugh. Y'all, this is the sickness that we're dealing with right now. Roundup, mold resistant. Something about eating that chemical, which then makes the mold grow more in you or something. It's hard to find, you know, the indie sources in Google. Oh, but this is fun, like organic replacements for Roundup. That's good to know about. You got oh, just take the weeds out yourself. Amen. Herbicidal soap and herbicidal vinegar. Products that contain 30% acetic acid kill weeds in just an hour. That's interesting. Has anyone tried that? The third option is citrus and clove oil contained in bonide burnout. You can see plant leaves wither as you watch, and the area smells good. Citrus and clove. I've heard about cayenne. I don't know if that's true. Ooh, this is in the AJC. Go AJC. I'm going to link it here in the chat. It's kind of cool. Hey, Jabri. Cool picture of your show today. So here, let's try this. The problem with Roundup seeds. I talk about this in my Hempdemic documentary as well, how DuPont family that could have been instrumental in removing cannabis and creating it as a controlled substance and taking hemp away from the people, making it controlled by corporations and the government, that family is in charge of selling a lot of our agricultural seeds. So I feel like agriculture is just this huge, it's this huge blind spot that more people need to just look at as seriously the core issue in society. It's what we're all centered around is eating. So we have to look at what we're doing when we're growing our food. Okay. So, whoa, this is really small writing. So Roundup Ready genes have been introduced into the food supply through cross-contamination, cross-pollination. For example, as stated in Greenpeace report, it's become virtually impossible for farmers to grow non-genetically engineered canola in Canada. Scientists don't know the true effect of these organisms on the environment before releasing them, and other issues stem from the idea that changing one gene does not necessarily change just the function. A variety of changes in the expression of such a gene could occur in the resulting organism. So this is a hot topic now, like changing the expression of genes. How do we do that? Well, this leads us to m6A. I'm not going to go there yet. But m6A changes the expression of our messenger RNA to not send out antiviral responses when our body would naturally do that. It's like the Trojan horse. It's like you have your defense, but how can you defend yourself if it's already inside you, leading those troops, which become your own body? So it's like if your enemy gets in charge of your army and says, attack her. And you're like, no, you're my army. Don't attack me. Attack the virus. But no, that m6A that's modified through a different expression causes your DNA to express differently so you don't defend yourself where you would. Now, GMO foods, I believe, cause this autoimmune issue that a lot of people have. Because let's look at this. When did we start using Roundup? And it might have been called something else. I should probably just say glyphosate. Okay. 1974, Monsanto developed and patented the use of glyphosate to kill weeds in the early 1970s and first brought it to market in 1974 under the Roundup brand name. So it was developing somewhere. So someone might have been getting sick where they were developing it. Now, when did aflatoxins show up? Okay, well, aflatoxins were discovered in the late 1950s and early 1960s when they were identified as causative agents of turkey X disease. What? An epidemic involving deaths of numerous turkey pouts, ducklings, and chicks fed diets containing certain lots of peanut meal originating in South America that were filled with aflatoxins. I'm definitely having some salad with some tuna and some broccoli and string beans and cucumber. Yeah, so it's kind of like a salad, but it's also sauteed. Good night, Jabri. Sleep super well. I'll see you tomorrow, Sunday Night Live. Okay. When did m6A show up? Um, the role of m6A modification in the biological functions and diseases. All right, I'm just going to drop this link right here. This was written in February 2021, this year. Anyway, we're not going to read that right now. In 1970, m6A was first recognized as an abundant nucleotide. That's just interesting. Very interesting. So apparently something like Shilajit with the fulvic acid and this like composty, supposedly that I hear helps to get the aflatoxins out of the body besides liver cleansing, because supposedly your kidneys and liver are attacked the most. Maybe your brain too, I don't know. Mold is just like not supposed to be in your body. It's not supposed to be in your house. It's not supposed to be in your body. It's not supposed to be in your food. Yeah. Talk to me, people. How are people exposed to aflatoxins? People can be exposed to aflatoxins by eating contaminated plant products such as peanuts, or by consuming meat or dairy products from animals that eat contaminated feed. Once again, farmers and other agricultural workers may be exposed by inhaling dust generated during the handling and processing of contaminated crops and feeds. Which cancers are associated with exposure to aflatoxins? Mainly liver cancer. Okay. All right. The World Health Organization, 2018, aflatoxins pose a serious health risk to humans and livestock. Aflatoxins, or poisonous substances produced by certain kinds of fungi and molds. This spells molds, M-O-U-L-D-S. I've never seen that spelling. That are found naturally all over the world. They can contaminate food crops and pose a serious health threat to humans and livestock. Aflatoxins also pose a significant economic burden, causing an estimated 25% or more of the world's food crops to be destroyed annually. And we're here worried about destroying hemp that's too high in THC. We should be more worried about the aflatoxins. Now, that's interesting, though. Most human exposure comes from nuts and grains. 25% or more of the world's food crops are destroyed annually because of aflatoxins. And how are they destroyed? And where do the aflatoxins go? Interesting, huh? Drought stress, insect damage, and poor storage can also contribute to higher occurrence of the molds, including in more temperate regions. There's definitely been mold issues in hemp that I've heard about. The high humidity is an issue, obviously. Okay. Okay, this is interesting. Long-term exposure can have serious health consequences. Long-term or chronic exposure to aflatoxins has several health consequences, including aflatoxins are potent carcinogens and may affect all organ systems, especially the liver and kidneys. They cause liver cancer and have been linked to other types of cancer. AFB is known to be carcinogenic in humans. the potency of aflatoxin to cause liver cancer is significantly enhanced in the presence of infection with hepatitis B virus. Huh. Aflatoxins are mutagenic in bacteria, so they affect the DNA. They're genotoxic, and they have the potential to cause birth defects in children. Children may become stunted, although these data have yet to be confirmed because other factors also contribute to growth faltering. For example, the low socioeconomic status, chronic diarrhea, infectious diseases, and malnutrition. Aflatoxins cause immunosuppression, therefore may decrease resistance to infectious agents. For example, HIV and tuberculosis. Now that's really interesting because in Hulda Clark's book, The Cure for All Diseases, have any of you read that? She talks about HIV being connected to a corn parasite. I wonder if the aflatoxins have something to do with it too. She said it attacks the thyroid. The parasite goes into your thyroid. And if you have a lot of benzene in your body, supposedly that's a parasite that causes like HIV symptoms or something. Isn't it wild? Like there's so many different perspectives of looking at disease. You can look at it as just the bacteria. Then what about the toxins in your body? I mean, there's so many mysteries to the world, you know? I'm getting full. Good thing I got my to-go container at this restaurant. Okay. Let's keep reading. Large doses of aflatoxins lead to acute poisoning that can be life-threatening, usually through damage to the liver. Outbreaks of acute liver failure, jaundice, lethargy, nausea, death, identified as aflatoxicosis, have been observed in human populations since the 1960s. Most recently, deaths attributed to aflatoxins were reported during the summer of 2016 in the United Republic of Tanzania. Adults are more tolerant to acute exposure than children. Why does acute sound so cute? The consumption of food containing aflatoxin concentration of 1 milligram or kilogram or higher has been suspected to cause aflatoxicosis. Based on past outbreaks, it has been estimated that when consumed over a period of 1 to 3 weeks, an AFB1 dose of 20 to 120 UG kilograms by weight per day is acutely toxic and potentially lethal. So AFB1 is what they're calling one of the toxins. Oh, it cooled down already. I wanted some hot tea. Hey, Diane. Shilajit! That's why you're so cheery all the time, isn't it? All right, I'm just about done with this food. But I like eating all the avocado and not saving it because then it turns mushy and brown the next day. And I won't want to eat it. This is not exactly the most ideal way to eat my dinner, like holding it up like a tray. This is a very interesting conversation, Diane. Do you know what aflatoxins and m6A are? Because I'm just learning, and it's a lot of terminology. But I think it's good to just start bringing this to the common dinner table and start chatting about it all. Because we're all of it. I think this is much more fun than being like, I'm Republican, you're a Democrat, you suck, I rock. Come on, people. Let's just all learn together. Let's break it down. Let's get geeky. All right, so let's talk about m6A for a minute here, because we have not talked about that enough. You know, I like Wikipedia because it kind of, if anything's going to break it down to you like you're a 2-year-old, I think it's Wikipedia. You know, I'm going to send you all the link. You can read along if you want. I think this is really, really interesting. N6-methyl adenosine. Now, adenosine, isn't that a word for a virus? Adenosine. Oh, it's an organic compound that occurs widely in nature in the form of diverse derivatives. Diverse derivatives? Is that like word salad? The molecule consists of an adenine attached to a ribose via a B and glycosidic bond. Adenosine is one of 4 nucleoside building blocks to DNA and RNA, which are essential for all life. Well, what are the other 3 nucleoside building blocks? Now I want to know that. There's only 4 building blocks in all DNA and RNA. We're getting down to molecular biology right now, which was not a class in college I enjoyed. As my teacher. Hope she's not watching. Doubt she is. But I just wanted to break down ATP and mitochondria a little more, and she wasn't having it. Anyway, now this is just saying 3 parts. Oh, the molecule's ribonucleotide building blocks are themselves made up of 3 parts. A sugar molecule. This is not the same as refined sugar, people. Don't think you're feeding your DNA by eating sugar. A phosphate group, and one of the 4 bases that form the alphabet of RNA's genetic code. Oh, so only one of these is in RNA's genetic code. Why not DNA? You didn't do well in biology. I'm sure you did better than you thought, Diane. Okay, so adenine. Oh, excuse me, that's not adenosine. Now it's saying adenine. Is that what it said before? Okay, adenosine, that's a glucose. Okay, anyway. Adenine, you're, okay. You're a cell, you're an imbecile. Sorry, I'm talking to myself. Cytosine, I don't mean it. And guanine. Oh, that reminds me of guanabana. I really like that fruit. Okay, I meant to say building blocks to nucleoside building blocks. So 4 nucleoside. Why is this even important? Like, why do I even want to know this? Why not? Don't you want to know what we're made of? Our DNA or RNA is made up of nucleoside building blocks. What the heck is that? Oh, now they're saying a nucleotide. I thought it was a nucleoside. They keep changing it up. One of them said nucleoside. One of them said nucleotide. Which side are you on? Do we want the ocean tide? Or what? There's Aviva in the house. Molecular Biology 101. If you want to fail your next test, watch this video instead of studying. All of you pre-med people out there over at Emory or Tufts or wherever you are. Okay. Oh, Garland's still here. You didn't even take biology. How dare you? But I bet you took astronomy. That's what all of us philosophers do. Okay. A nucleotide is the basic building block of nucleic acids. How come we stop talking about nucleosides? I like that word. Physics, but not astronomy? How could you not want to look at the stars? See, now that's saying adenine too. It's not saying adenosine. Anyway, adenine, cytosine, guanine, and thymine. Who cares? We are the soul. Adenine, thymine, guanine, and cytosine, cytosine. All right, molecular biology geeks, this is your song to study for your next test. They didn't have astronomy when they were like so serious. They didn't want you to do anything fun. Adenine, thymine, guanine, and cytosine. So when you look at these like advanced science journals, they've got abbreviations for each of those nucleotides. A, T, G, and C. Adenine, thymine, guanine, cytosine. It's like a cytostorm. Cytosine. Oh, I love you too, Diane. Thanks for hanging out. I should probably go soon too, but I gotta finish some food. I'll see you tomorrow, girl. I hope. I love you. I want to know. I feel like there's an adenosine virus. I want to know what that is. Is adeno-associated virus? And there's an adenovirus. It's used in gene therapy and as vaccines to express foreign antigens. Adenovirus vectors. Maybe that's where I saw the word in the shot ingredients. Can be replication defective. Certain essential viral genes are deleted and replaced by a cassette that expresses a foreign therapeutic gene. Such vectors are used for gene therapy, as vaccines, and for cancer therapy. What are we, computers? I don't understand. So adenovirus vectors. AAV, that's something else, are non-enveloped icosahedral viruses with a single-stranded DNA genome. An infected individual or through direct contact with the contaminated environment. Is AAV a DNA virus? This is wild, isn't it? So much information out there. What is the maximum length of DNA that can be inserted with adenovirus? This is so weird. The amount of DNA that can be effectively packaged into adenovirus virions is about 105% of the wild-type genome, and thus allows for the insertion of about 2 kbs, it couldn't be kilobytes, of foreign DNA. With the E1A deleted viruses, it has been possible to insert 5 to 6 kb of DNA. I apologize, I'm not a doctor. I don't know what I'm talking about right here. And I know we shouldn't be Googling stuff like this, but Google exists. It's at our fingertips. At least I talk to mentors too, unlike some millennials. But I'm not criticizing you. So what's an adeno-associated virus? Oh, AAV. They're small viruses that infect humans. So what's corona? Is corona an adenovirus? Am I full of Shilajit or what? No, it's not. All right. Your Medical Imaging Society has some pictures here. This is just interesting. I'm going to let you all look at this if you want. We got a little bit off track here. I want to know everything. I just want to know it all. But wisdom is not necessarily having to know it all, just knowing how to apply the knowledge that you do have. But yeah, anyway, I knew I heard the word adeno in the word virus, so. Ay, ay, ay. What's in a name? Sometimes it's better to just not know, you know? Okay. N6-methyladenosine, m6A. Why isn't it called N6A if it says N6-methyladenosine? I guess because the methyl and the adenosine is more important than the N6. What the heck is the N6? Anyway, methyl-6-denosine was originally identified and partially characterized in the 1970s. There's 4 sources there. And is an abundant modification in mRNA and DNA. It is found within some viruses. And when it is found within a virus, people, this is what I read, that virus is more deadly. Because when it's found in it, it's telling your DNA to not defend itself. And then you get gobbled up by the virus. And then you don't have antiviral armies. And supposedly there's a couple of strains of SARS-CoV stuff. And some of them have the m6A and some of them don't. So guess which one is a little bit not fun to get? Supposedly the one with the m6A, right? The methyl-6-adenosine. Anyway, it is found within some viruses, and most eukaryotes, it probably is pronounced eukaryotes. Most eukaryotes, including mammals. What the heck is a eukaryote? Okay, we gotta look up how to pronounce this thing, because that is just the coolest word in the whole dictionary. And y'all, if you're playing Scrabble out there, EUKARYOTE. Can I just cut and paste from Wiki? I think I can. Bam. Wiki, wiki, wiki. Is Wiki in Hawaii? Eukaryote. That's how you say it. It sounds like a Hebrew word. Eukaryote. Hey, Jordan, what's up? Well, thank you for the like. Thank you for the like. That was so nice of you. Okay. Eukaryote. That is my new favorite word. Eukaryotes, or organisms whose cells have a nucleus enclosed within a nuclear envelope. That's so cute. That's like the post office inside of our bodies. They're sending emails to each other. No, they're old school. They're sending letters. They have envelopes. Eukaryotes belong to the domain eukaryota, or eukarya. Their name comes from the Greek eu and kapiov. Eukaryote is Greek. I guess Greek kind of sounds like Hebrew. Yeah, Middle Eastern. Why are you so tired, Jordan? Oh, DC. Anyway, what's a eukaryote? It's an organism consisting of a cell or cells in which the genetic material is DNA in the form of chromosomes contained within a distinct nucleus. Eukaryotes include all living organisms other than the eubacteria and the archaebacteria. We are all eukaryotes. We are one eukaryote race. It really should be eukaryote. That would be so cute, because it's got an E at the end, but it's just eukaryote. That's okay. It sounds like you carry out your mission, because you are a living organism. You carry out. You carry out. You carry out the trash today. Because you're a living organism. You're a eukaryote. What are you? What are you? You're a eukaryote. That's what you are. You're such a cute little eukaryote. I just want to squeeze your cheek. Eukaryote, you're such a beautiful baby. And my poor Pacey is running out of batteries. What are we going to do with you?
Hello, baby. Let's go kissing.
See, I'm starting to distort. I look smarter like this. Tommy!
I love playing with you, baby.
Have you heard of Shilajit, Tommy? Have you heard of a m6A? It's not a gun. Have you heard of aflatoxins? I'm sure you have at least heard about aflatoxins. I don't know about the other Shilajit that she's talking about. But you are a eukaryote. Did you know that, Tommy? What's my point here? How long have I been going? Oh my God, it's over an hour. Good. I have a social life. Thank God. Okay. I have smudgy on my glasses. It is time to put some of my food in my to-go container to protect it from aflatoxins. Are we laflatoxins or are we aflatoxins? I think we need to laugh a lot together. I'm stuck in the prison of my own little seat and I don't want to get up. All right, so what I'm trying to say is there's too much information. Most eukaryotes, including mammals, insects, plants, and yeast, is where the abundant modification in mRNA and DNA, albeit m6A, is found. m6A is a modification of messenger RNA. We got to talk about modifying things. Now, I think you can have good modifications. Like, I believe in the therapy that I teach about self-awareness. I believe that my DNA or my RNA is changing because the DNA is like, the DNA might not change. Okay, the DNA is our blueprints. Okay, I mean, I've known crazy things to happen. Like, I've known people who change their eye color to blue because they meditate on it every day. You know, I mean, it's ridiculous in a way. I love JP Sears' video of like the guy's like, I'm meditating hard on a Ferrari being in my driveway right now. Is it there yet? Is it there now? Is it there now? But anyway, with patience, I do think we do have manifesting powers that are a little more realistic. But what was I just saying? I was talking about, what was I just talking about? The many tangents of my weird brain the past couple of days. What was I talking about? Eukaryotes. Oh my goodness. Modified. Oh yeah. So I believe when we go into our inner child work, which I love doing, it's hard work, but so is making dinner. And you get to eat it at the end of making it, and you're like, wow. So when you do this inner child work and you like go back to a memory, like what memory was I working on? I'll just share it with you. What memory was I working on yesterday? Probably something traumatic that I might not want to share. I don't even know. But let's say I go back to a memory of like doing a creative project when I was in elementary school and getting into a fight with my best friend who decided, I'm not going to be in your project anymore. And then she leaves me stuck having to sing and play the xylophone at the same time and act like the genie and the little girl in bed at the same time. So she left me with this impossible task last minute. It's only an 8-6-second video clip from the MSM of Down Under. Whenever you have time to check, send me the link. Send me the link, Tommy. Put it right there in the comments so I can just copy and paste. I see you did the topic, but what if it doesn't come up? Anyway, so little Aviva was like, why did I get abandoned? There must be something wrong with me. Like, was I mean to her? Like, she didn't tell me I was mean to her or anything. Like, it was just one of those things. I didn't know if I did anything wrong. I didn't have any parents I had parents, but I didn't have parents that I could talk to about this for some reason, like, or they didn't think it was important. I mean, it's just a child's not important little drama, apparently. You're not Vec. You're not tech. All right, let me see if it comes up with just this, Tommy. Anyway, I see 2-hour videos coming up. Jam noise. Anyway, I did drag it into the search, and there's like 20 options. I don't know which one it is. So as I was saying, when I work through it in the process that I'm doing, it's like this untangling effect, like untying of a knot, so that I can see myself, and process it in a slow way, slowing down, being the parent to myself, being the friend to myself. And when we have experiences in childhood, it really imprints on us. Like we're water, and there's like a picture gallery in us. And so when you go through that and you change your perspective, then you literally kind of have new photos to add to the album. So it would make sense that maybe the RNA changes in you too. Maybe there's like a little different messaging sequence that goes on in that inner post office of communication. Get it? Got it? Good. Okay. So we're talking about modified RNA. And so this abundant modification of the messenger RNA is also found in tRNA, rRNA, and small nuclear RNA, as well as several long non-coding RNA, such as exist, XIST, exist, we exist together. An hour later, and you're still with me. Am I going to tie this up to some kind of conclusion? I don't think so. I think I'm going to leave you up to just keep digging in this soil until something beautiful grows out of it. You know what I'm saying? That's all I want. Just a continued conversation so we can grow daisies and sunflowers in the Shilajit. Because I don't like sitting in a pile of Shilajit. I at least want some flowers. Oh my goodness. Let's talk a little bit more about m6A. Or not. All I have to say is, in one of the studies I looked at yesterday, there was more modified messenger RNA saying for the viruses to stay and the antiviruses to stop fighting the viruses in the body when the aflatoxins were also present in the body. So that's the connection that I found in one of those PubMed studies. Yeah. Excuse me. Mapping of m6A in human and mouse RNA has identified over 18,000 m6A sites in the transcript of more than 7,000 human genes with a consensus sequence of blah, 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 consistent with the previously identified motif. 18,000 m6A sites in the transcripts of more than 7,000 human genes. The localization of individual m6A sites in many mRNAs is highly similar between human and mouse. And transcriptome-wide analysis reveals that m6A is found in regions of high evolutionary conservation. m6A is found within long internal exons. Is that where that gas station got its name? Exons? And is preferentially enriched within 3 UTRs and around stop codons. I'm glad you got away, Tommy. What the heck's an exon? Is it a tail of the DNA thing? Not to be confused with an axon. That's right. It's been a few years since molecular biology for me. Okay, not to be confused with an axon, the projection of a neuron, or exon, a brand of fuel, or hexon, a viral protein. That's funny. An exon is any part of a gene that will encode a part of the final mature RNA produced by that gene after introns have been removed by RNA splicing. The term exon refers to both the DNA sequence within a gene and to the corresponding sequence in RNA transcripts. In RNA splicing, introns are removed and exons are covalently joined to one another as part of generating the mature messenger RNA, just as the entire set of genes for a species constitutes the genome. The entire set of exons constitute the exome. Are you a genome or an exome? That's the question. Where was I? Happy to hear. What did you have for dinner tonight? Anyway, for some reason last night I was obsessed with learning as much as I could about m6A because it's been reported to impact viral infections, and that's a hot topic right now. So, if m6A is present in us already and we're attacked by something like a virus, that might mean that we're higher risk for, I don't know, not recovering as well. Don't you think? We're getting sicker. So, m6A has been reported to impact viral infections. Many RNA viruses, including SV40, adenovirus. See, I knew I read about adenovirus. Oh no, I was looking up something else. Okay, adenovirus, herpes virus, Ralph-sarcoma virus, and influenza virus have been known to contain internal m6A methylation on virus genomic RNA. So m6A methylation is the methylated thing that stops you from fighting off viruses. Several more recent studies have revealed that m6A regulators govern the efficiency of infection and replication. Why do you have to say it like it's a good thing? Like your infection is efficient. We don't want the infection to be efficient. Anyway, I know what you're trying to say. Efficiency of infection and replication of RNA viruses, such as human immunodeficiency virus, HIV, hepatitis C, which is HCV, Zika, which is ZIKV, and these results suggest that m6A and its cognate factors play crucial roles in regulating virus life cycles and host viral interactions. So, can we just get rid of the m6A in our own body? Thanks for hanging out so long, Garland. I'm about to check out too and finish the song for tomorrow. Oh, Garland, did you want to invite people to the event tomorrow? Do you have a link or anything? I think it's a fundraising event. Okay. So now I'm going to Google, can you remove methylated m6A? Or how about, can you decrease in your body? But see, this is like a fuzzy area because I think there are some biological functions that need m6A. It has a role, I think, in the nervous system, but maybe not when it's methylated. And I think they were also connecting it with obesity. It epigenetically impacts biological processes. And it's a reversible post-translational modification to RNA. All right, well, here's a hot topic, cardiovascular disease, right? This is from Spandido's Publications, Internal Journal of Molecular Medicine. Check the source, check the source. Love you. All right, this is it, and then I'm going back to work on some songs. N6-methyladenosine, or M6A, is the most prevalent and abundant type of internal post-transcriptional RNA modification in eukaryotic cells. Multiple types of RNA, including mRNA, rRNA, tRNA, long non-coding RNAs, and macroRNAs, are involved in M6A methylation. The biological function of M6A modification is dynamically and reversibly mediated. Thank goodness there's a mediator in the room. By methyltransferases. They're the writers in your body. What, you think they're going to write their own letters? No. They're rich. They got writers. Demethylases, erasers, if they're going to write, they need erasers too. And M6A binding proteins, which are the readers. It's like a whole village in there. The methyltransferase complex is responsible for the catalyzation of M6A modification and is typically made up of methyltransferase, like METTL3, METTL14, and Wilms tumor 1 associated protein. Erasers remove methylation by fat mass and obesity associated protein and ALKB homolog 5. Readers play a role through the recognition of m6A modified targeted RNA, the YT521B homology domain family, heterogeneous nuclear ribonuclear protein, and insulin-like growth factor 2 mRNA binding protein serve as m6A readers. The m6A, it's like everyone, you got the cast of characters here. We're about to do the reading of the play. All right, we just got to assign all the roles first. The m6A methylation on transcripts plays a pivotal role in the regulation of downstream molecular events and biological functions, such as RNA splicing, transport, stability, and translatability at the post-transcriptional levels. You know, because one molecule might speak Spanish, the other one might speak French. We got to translate the languages to each other. The dysregulation of m6A modification is associated with cancer, drug resistance, virus replication, and the pluripotency of embryonic stem cells. Recently, a number of studies have identified aberrant m6A methylation in cardiovascular diseases, CVDs, including cardiac hypertrophy, heart failure, arterial aneurysm, vascular calcifications, and pulmonary hypertension. The aim of the present review article was to summarize the recent research progress on the role of m6A modification in CVD, cardiovascular disease, and give a brief perspective on its prospective applications in CVD. Sounds interesting to me. Did I give you the, yeah, I posted the article right there. All right, let's skip to the conclusion. Oh my goodness. Oh gosh, this is so interesting, but I'm getting too tired. They talk about the m6A sites. That's interesting. Epigenetic research. This sounds scary. Why do we need to do this? There is an emergency evidence to indicate that m6A modification is closely related to the occurrence and progression of cardiovascular disease. Okay. Including cardiac hypertrophy, heart failure, ischemic heart disease, aortic aneurysm, vascular calcification, pulmonary hypertension, etc. Herein, the recent progress in m6A modification in CVD is briefly reviewed. Okay, so I feel like since sugar is a big indicator for, like, it causes a lot of cardiovascular disease, I wonder if sugar causes the m6A modification to begin with, or corn syrup. Like, why is it there? And why is it causing the heart disease? That's just interesting stuff to me. Like what they see, the ingredients, you know? It's like someone comes into your house and they see the sink and they're like, okay, the sink is there. And then they see you're making dinner and they're like, oh, the pans are there. And it's like, and then all of a sudden there's a robber there. They're like, oh, there's a robber there. What caused the robber to be there? Maybe it was the sinks and the pans. Very weird. Okay, how's the conclusion? And then it's time to say goodbye. Conclusion and future perspectives. m6A methylation is a type of RNA epigenetics and the most abundant internal RNA modification type. What do you mean by epigenetics? The type of, let's break that down. It's just the study of heritable changes in gene expression. So it's a change in the gene expression. So it's a type of, so m6A methylation is a change in your RNA genes expression. Okay, m6A methylation. What causes this? Okay, I'm gonna just look that up before I say goodbye and see if there's any studies on what causes m6A methylation. Oh yeah, I did find that last night. There was a bunch of things like heavy metals and all. Anyway. Okay. and it's the most abundant internal RNA modification type. M6A methylation is an emergency research field that's growing rapidly, especially in cardiovascular disease. The present article reviewed the role of m6A and m6A SNPs in the mechanisms of various types of CVD. The observations highlight the potential role in the diagnosis of CVD. Oh, okay, by serving as an invasive biomarker and lay the foundation for the accurate treatment of CVD. So now they're saying they'll test you to see if you have m6A methylation in your body so that you know if you're at risk for cardiovascular disease. But I think if you have that, you're at risk for everything because it stops you from killing viruses too. Hey, Frank. Ew. I am not answering that question. Okay. Anyway, m6A methylation presents across different species and plays a fundamental role in cardiac biological processes and the pathogenesis of CVD. An increasing number of studies have uncovered the novel dysregulation of m6A methylation as a hallmark of the development of CVDs. So they're calling it a dysregulation of m6A methylation because I guess it's somewhat regulated otherwise. Oh, this is so confusing. The dysregulation of methyltransferase, demethylases, and m6A binding proteins play a role in the occurrence and progression of cardiovascular disease. Studies have been demonstrated that the regulation of these writers, erasers, and readers may alleviate the progressive of cardiac disease. I think what they're trying to say, see, they need to write this a little bit better. The novel dysregulation because of m6A methylation. So m6A methylation modifies the RNA so that it stops regulating the writers, erasers, and readers. So if it's being regulated, it's saying it alleviates the progression of cardiovascular disease. And so it can be a diagnostic biomarker. Okay. What causes m6A methylation? They don't say what causes it. They don't know. I think it's eating sugar. I really do. Yeast, candida. I think that's what causes it. Yeast and candida in the body. Why does someone have m6A methylation? I mean, it just sounds like an alcoholic cell. Methylation. All right, one more. Okay, let's look at the 10th page. Let's see what's hidden there. Nothing. 6th page. Dong Tao chicken genetics. M6A methylation. What about this? Caused by man-made lab. What about that, you guys? That's scary. Nah, no one's done this study. But it's saying, okay, here we go. March 22nd, 2021. That was like less than a month ago. m6A RNA methylation facilitates pre-mRNA's 3' formation and is essential for the viability of Toxoplasma gondii, a plant-like mechanism coupling m6A reading to polyadenylation safeguards transcriptome integrity and developmental genes partitioning in toxoplasma. Toxoplasma gondii, or eukaryotic parasites that can infect any warm-blooded animals, including humans. In fact, it's thought that 1/3 of the human population is infected with toxoplasma parasites. In most cases, the infection is benign, but it can cause serious health hazards to immune-compromised individuals and can cause congenital birth defects. Currently, there are few therapeutic options to treat toxoplasmosis. In these two preprints, Holmes, blah, 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 discover important pathways for messenger RNA processing and stability in Toxoplasma gondii parasites that might serve as a new therapeutic target for anti-toxoplasma intervention. This parasite possesses a complex life cycle inside mammals. Humans are infected by either ingestion of infected meat or from contact with infected felines. As the infection progresses, parasites differentiate into cysts, namely in the brain, or bradyzoites. These forms are quiescent and cannot be targeted by current therapies. Transitions between cellular stages, such as in differentiation, are increasingly associated with regulation at the mRNA level, in addition to transcriptional changes. As the case with DNA, mRNA can also be modified that affects its stabilities, localization, and translation. mRNA ramifications, or the epitranscriptions scriptome, seem to be present in eukaryotic kingdoms, and its role in cellular processes is still being unraveled. The most common modification is the methylation of adenosine residues, termed m6A. So now you're saying m6A are adenosine residues. Hey, Dusty. Termed m6A. Nonetheless, some proteins have been identified as m6A writers, readers, or erasers, depending on the function they perform on the epitranscriptome. Adenosine residues. That sounds like the adenosine popped out of the RNA, like the nucleotide popped out of it. I mean, maybe it popped out to try to bind with the sugar in the body. You know what I mean? These two preprints look at m6A methylation in toxoplasma parasites from different approaches. On one hand, Holmes aimed to understand how this modification might help the transition between the active and the quiescent forms of the toxoplasma parasite. These authors focused mostly on the m6A writer protein, METTL3. On the other hand, Farhat et al. Focused on the reader protein, namely the CPSF complex, that seems to have functions that are shared between toxoplasma and the plant Arabidopsis thaliana. Anyway, blah, 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 blah. Are we done here for now? Oh. Okay, hello. Eureka alert. All right, pub releases. Here we go. Chemical modification of RNA could play a key role in polycystic kidney disease. A chemical modification of RNA that can be influenced by diet appears to play a key role in polycystic kidney disease, an inherited disorder that is the fourth leading cause of kidney failure. The findings published online today April 13th, 2021. See, I am trending. Suggests new ways to treat this incurable condition. Blah, 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 blah. The researchers investigated whether the most common RNA chemical modification known as m6A, which occurs when a methyl group is chemically attached to an RNA component called adenosine. Okay, so in everything I read for hours last night and today, it still was not explained to me that a methyl group gets chemically attached to the adenosine. Okay, so his whole sentence is, the researchers investigated whether the most common RNA chemical modification, known as m6A, which occurs when a methyl group is chemically attached to an RNA component called adenosine, might be altered in PKD. I forget what PKD is. Specifically, they looked at the activity of the writer, the enzyme that performs this methylation reaction. In 3 mouse models of PKD, they found, is that kidney disease, PKD? They found that the resulting m6A levels were significantly higher in healthy animals without this condition. The same was true for kidney samples from PKD patients compared with healthy kidney samples. Higher in healthy? No, wait, this doesn't sound right. Yes, you want some, Dusty? Here, I'll put it in a to-go container for you. Yeah, I have a gig in Smyrna tomorrow. Whoops. Yeah, how are you doing, Dusty? It's been a while. I'm going to eat the avocado because I don't think that'll stay good. Let's see how much I can get in here. I need like a spoon. It's working. All right, let's see if I could read at the same time and not spill it all over the place. See, this is what's confusing. Like, is m6A good or bad? Well, it's modification. I mean, it's like meditating on things and changing them in the moment, you know? I got it on my sweater. That's what happens when you do so many things at once. But that's why I got a napkin. Holy moly. Okay. Further investigation showed that in mouse models, metal-3 activity increased before the first kidney cysts made their appearance, suggesting that it might be an initiating event for the disease. Interesting way to say it. Don't fall! Speaking of mouse, on my mouse. When researchers genetically altered healthy mice to overproduce metal-3, oh, that's so mean, the animals developed small kidney cysts, even though they didn't carry any PKD mutations. Conversely, shutting down metal-3 in the PKD model significantly slowed the cyst growth. Okay. Suggesting that the enzyme plays a key role in disease progression. Makes sense. Next, the scientific team tested whether limiting methionine, the dietary nutrient that supplies the raw materials for methylation, might stem cyst formation. When researchers grew kidney tissue in petri dishes with varying concentrations of methionine, cysts increased with higher concentrations. Okay, so methionine causes the methylation. Okay, so now we know what causes the methylation. Let's look up what has that in it so we can avoid it, right? The researchers saw an opposite phenomenon when PKD mice were fed a low methionine diet. These animals had less severe PKD. All right. I think a low methionine diet is the answer to prevent a lot of disease. Doesn't it sound like that? What the heck is that? Ramalingam notes that the findings have two important implications. First, it may be possible to partially control PKD with a vegan or vegetarian diet, since methionine is found in meat and fish. Well, I just had a bunch of methionine. I'll let you know how I feel tonight. That is really fascinating, y'all. You sure you still want this, Dusty? Meat and fish. But is it polluted meat and fish? Is it like mercury-laden fish and heavy metal meat? Second, identifying chemicals that stem metal activity may lead to new drugs to treat this condition. All right. Wow. Okay. What did you say, Dusty? Busy as heck. Aw. Okay. Bye. Let's look up what causes the methylation is the methionine. I love how I can just copy and paste from Google. Thionine. That is our, what do you call it? The bad guy. It's an amino acid, and it's the building blocks that our bodies use to make proteins. Okay. Methionine is found in meat, fish, and dairy products, and it plays an important role in the many functions within the body. Okay. Except it also can be an issue if you have kidney issues. Interesting. But like, what is it? What is methionine? Sounds like iodine. Methionine. Now they're saying they give it to you. This is confusing. Now they're saying they give it to you as medicine to treat liver issues and viral infections. However, if it creates more methylated m6A, then we shouldn't be taking it. Right? I'm confused. Man, I hate when food gets stuck in that little area. Oh well. In acetaminophen poisoning, methionine prevents the breakdown products of acetaminophen from damaging the liver. Interesting. It may also act as an antioxidant and help to protect damaged tissues. Weird. Yeah. That is really strange. That's from WebMD. All right. This is my brain. Okay. Methionine, or methionine, it doesn't tell me how to pronounce it, is an essential amino acid in humans as the substrate for other amino acids such as cysteine and taurine, versatile compounds such as SAM-E, and the important antioxidant glutathione. Methionine plays a critical role in the metabolism and health of many species, including humans. It's encoded by the codon AUG. Freaking weird. Methionine is also an important part of angiogenesis, the growth of new blood vessels. Supplementation may benefit those suffering from copper poisoning. Overconsumption of methionine, the methyl group donor in DNA methylation, is related to cancer growth in a number of studies. So if you overdo it, that's the m6A modification issue. Okay. Methionine was first isolated in 1921 by John Howard Mueller. All right, so can we please see everything that methionine is in so we don't overdo it and get cancer? What is methionine in? It's used in lipotropics, lipo injection. Okay. Just meat, fish, and dairy. That's all you got for me? Come on, can't you break it down? So methionine's not in any vegetables? It's in dairy? Okay, how much methionine is in dairy? There we go. Top 10 foods highest in methionine. Okay. So everything's a balance, people. If you have a deficiency, it causes liver issues, anemia, and graying hair. And if you have a low, if you do have low methionine, you have an extended lifespan and reduce the risk of cancer. Turkey, beef, fish, pork, tofu, milk, cheese, nuts, beans, and whole grains like quinoa all have methionine. Okay. So just eat vegetables and fruit, basically. The recommended daily intake is 10.4 milligrams per kilogram of body weight. Okay, below is the list of the top 10 foods highest in methionine with the calculation. Okay, so ground turkey. So people who think that, people who are like, I'm going to have a turkey burger instead of a beef burger because I'm going to be healthier, they're getting dangerous levels, possibly, of methionine, which causes this m6A modification, which can cause obesity, cancer, blah, blah, blah. Not attacking the viruses appropriately. Okay, so so ground turkey has, and this could be because of the GMO stuff I want, that I was thinking about with turkey. For 6 ounces, you get the 207% RDI. So is that part of why you get so tired after you eat turkey on Thanksgiving? Yeah, if you have 100 grams, you have 128% of your daily intake that's recommended. And if you only have 200 calories of it, then you still have 169%. Okay, wait, that was more, actually. Okay, so more poultry high in methionine, 195% in 6 ounces of chicken breast. Dang. So there's more in chicken than beef. 93% in a drumstick, 40% in a chicken hot dog, 115% in a chicken thigh. So y'all, you don't need to be chowing on that much meat. That's really interesting. Like, just a little bit at a time, right? Okay, so a steak, a 6-ounce steak, and you've got 211% of methionine. Dang. 100 grams, you've got 124%. 200 calories, you've got 93%. If you've got a beef stew for 3 ounces, you've got 110%. If you've got lamb roast, you've got 106%. If you have veal, 100%. And if you have buffalo steak, you have 89%. So buffalo steak's better for you as far as methionine. Tuna! That's what I ate tonight. Oh my gosh, y'all. This is insane. What the heck is it? Methionine? Is it just natural or is it like, anyway. So in a 6-ounce filet, which I had at least one of tonight, 207%. Oh no. 100 grams, 122%. 200 calories, 132%. 200% for a 7-ounce grouper, 200% for a 6-ounce salmon. So actually salmon has more than tuna. 182% for snapper, which also has, oh no, wait, tuna's a little bit more actually than salmon. 7% more. Tilapia has 179%, but it's kind of GMO. Mahi. Actually, that's what I had, mahi tonight, not tuna. So I'm a little bit better, 153%. Lean pork chops are still over 100%. Everyone 198% for 6-ounce chops. 106% for lean ham. And then we go down to 3 ounces of salami is just 55%. And actually 3 slices of bacon is just 46%. But I think you probably have more than 3 slices. Okay. So firm tofu is also high in methionine. It's 1 cup has 73%. 100 grams has 29%, and 200 calories has 40%. Oh! Boiled soybeans, edamame, has 53%. Cooked soybean sprouts only has 13%. And soy milk, a cup, has 9%. Milk. So milk has 59% for a 16-ounce glass, 12% for 88 milligrams, 71% for 518 milligrams, and 57% for a cup of yogurt, and 29% for a cup of buttermilk. Low-fat ricotta cheese, also for just half a cup, it's 48% of your recommended daily. For 412 milligrams, it's 57%. And then we've got grated Parmesan. Per ounce is 36%. For Gruyere cheese is 32%. Swiss per ounce is 31%. So you got a couple ounces of Swiss cheese and you're at 60%. Per ounce of Gouda is just 28%, and Fontina, 28%. Okay, Brazil nuts, y'all. I've been kicking the butt on some Brazil nuts because of the selenium, but here you go. A handful has 40% of the recommended daily intake of methionine. Okay. Oh my God. Hemp seeds per ounce, 36%, y'all. So maybe that's why you need to slow down. Maybe CBD, we need to be checking our CBD oils for methionine content because we don't want high methionine. That can mess up our m6As, our RNAs. That could be some epigenetic issues right there if we're having too much of the oil at once while having meat and everything. Dang, 36% per ounce of hemp seeds. Oh my God. 26% for pumpkin seeds per ounce, 23% for chia seeds, 22% for toasted sesame seeds, 16% for sunflower seeds, 14% per ounce of flax seeds, 14% per ounce of cashews, 14% per ounce of pistachios, 11% per ounce of peanuts. So 2 more large white beans. Interesting. So I guess that's like baked beans. Per cup, 36%. Navy beans per cup, 35%. Kidney beans, 35% per cup. Black beans, 31%. Northern beans, 30%. Pinto beans, 27%. So this is like the #1 reason to cut meat out of your diet, the methionine, which is probably why it's really related to cardiac issues even more than fat. Quinoa. has some methionine issues, like a lot of grains. 24% per cup. For teff, 43%. For wild rice, 27%. Kamut, 23%. Rice, regular rice is just 14%. And spaghetti is 11%. All right, that's it, people. But what I'm wondering is, okay, here's some more. Shrimp, 78% per 3 ounce. Clams, 67%. Eggs, only 27%. Green peas, 18%. Sweet potatoes, 17%. Avocados, 10%. Kiwi fruit, 6%. You can get it from like anything. Mammy sapote, those are good, 6%. Crab has 100% in a crab leg. So what the heck? What the hecky wacky? Quinoa. All right. So, are we learning something? Everything in moderation, people. That's the lesson. Can you do that? Are you all addicts? No. So L-methionine is what it really is. It's used for protein synthesis, including the formation of L-cysteine, taurine, L-homocysteine, and sulfate. That is so interesting. Oh my goodness. So you do need to eat some of it, along with histidine, isoleucine, leucine, lysine, phenylalanine, threonine, tryptophan, and valine. Oh my gosh. That's so interesting. I wonder if cysteine helps it break down, like if you have too much of it. All right, I'm almost done, people. I can't believe I'm still alive. Cysteine. What food has cysteine? I feel like cysteine foods might be good for you. High protein. Anyway, tell me what y'all think about this. It contains sulfur, and it's a component of the antioxidant glutathione. Top 10 foods high in cysteine: lean pork chops, beef, chicken breast, tuna, lentils, oatmeal. I mean, that's a lot too. Tuna has 190% for 6 ounce filet. Oatmeal has 79%. Wow, oatmeal has it. Eggs have it. Low fat yogurt has it. Sunflower seeds, 38% per handful. That's a lot. Okay. All right, we are done here, people. I love you. Thanks for hanging out. Ooh, I got 3 thumbs up so far. There's 3 for you. Oh, is that my tummy digesting? Did you just hear that? All right, beautiful people. Ooh, I got another thumbs up. Thank you. All right. What did we learn? It's like the more we know, the more we don't know, right? Trust your instincts. That's what we learned. I've got a really cool Qigong class coming on Monday. I did this amazing preparation for it today. I'm so excited to share it. Anyway, I'll see you beautiful people tomorrow, Sunday Night Live. Feel free to shoot me requests musically, and I will do my best to have it ready for you. And give yourself a hug for me. My tummy just said she loves you too. And I will see you next time.